Okay, so we're going to talk about combined anatomy and physiology. When we're talking about anatomy and physiology, we're talking about anatomy as representing the form and structure of the body and its parts, and physiology talks to the functions of the body and its parts. And anatomy, of course, has implications for physiology. So if anatomy is not working correctly, then the physiology often can't function the way it should either. Perfect example of this is my old dog Nelson. He was just a puppy. We got him at about 4 months old, and by about 9 months old we realized that there was something wrong with him. He was urinating a lot. So this little guy was born with a malformed kidney. So with a malformed kidney, that's the anatomical component that's incorrect, not formed properly. But because the kidney is malformed, it creates challenges for the physiology to function properly. So this little guy ended up going into full-blown kidney failure because his kidney was malformed. The physiology also got all messed up and wasn't able to function as normal. So the approach to anatomy and physiology, I'm going to look generally at a systemic approach, but we will start just with baseline, individual areas, and then look at the broader system as we go through. Looking at each component of anatomy and physiology as part of a system is going to be the goal. And a good example is the urinary system, discussion of the structures and the functions of the components of the urinary system. So rather than just looking at the bladder or just looking at the kidneys, we typically like to talk about them all as part of a systemic approach. So we'll start by talking to some directional terminology. I'll go over this very briefly because there are a lot of resources for this, as well as the text, and I have another PowerPoint video that really starts off with directional terminology. So when we're talking about directional terminology on dogs and cats, it's important to get to know it because it gives us a universal language to identify where on the body we're acknowledging something. So if there's an area that is a concern, if there's something wrong with an area, like a tumor, a growth, a laceration, then we need to describe it in terms that are universal so that when the doctor picks up the medical record, they understand what we mean as to where this wound or injury is. If we created our own language all the time, there'd be a lot of miscommunication about where things are located on the body. So that being said, we typically divide the body into the median plane, which is through the middle here, and that creates a left and right side. We also divide the body through the transverse plane, which in this case creates the head and the tail end. And then, of course, the dorsal plane, which creates the top of the dog, or the dorsum, and sort of the bottom of the dog, which is the ventral aspect. So examples we went through in class, if we're looking at identifying wounds or areas on an animal, looking at directional terminology. If we're talking about on the body toward the head, then we're talking about cranial. If we're talking about on the body toward the tail, we're talking about caudal. If we're focusing on the head, we can't say it's toward the head if it's on the head. So instead, we would identify if it's more toward the nose, which is rostral, or more toward the back of the head, toward the tail, which is caudal. If we're talking about the dorsal plane, which splits the dog into like a top and bottom, we've got dorsal is toward the spine, and a good memory trick for that one is thinking about a dorsal fin on a dolphin. So dorsal is toward the spine of the animal. And if we look at the animal's belly, that's ventral. So ventral is toward the belly of the animal. And then we get into medial and lateral, which is a little bit difficult to appreciate on this image because it's just a 1D image. But we're talking about the midline. So if you put your hand at your belly button, essentially that is your midline. And that runs all the way up your body and all the way down your body. So anything that is on the body toward the midline, then we'll call that medial. So toward the middle of the body, that's medial. And toward the outside of the body, so the side of the ribs, the side of the abdomen, the side of the leg, that's all called lateral. So we've got medial is toward the midline, and lateral is away from the midline. When we're talking about limbs, if you look at his front limb there, of course we'll talk about left and right, but when it's closer to the body, So we split the limb essentially in half. And if it's closer to the body, we call that the proximal aspect. If it's further away from the body, we call that the distal aspect. And a little bit more advanced. This one gives a better perspective about medial and lateral. Still kind of challenging. But essentially, down through the center of the dog is the medial aspect. And then, of course, out to the side here is the lateral. And then we have, when we're talking about feet, we have to either be talking about front feet or back feet. So front feet, if we're talking about something that's on the top of the foot, same with the back feet actually, we call that the dorsal aspect. So the top of the foot is the dorsal aspect. And then the bottom of the foot where the pads are, if it's the front foot, we call that palmar. And if it's the hind foot, we call it plantar. So that's where the pads are on the underside of the foot. Now, a little memory trick, palmar, front feet. If you think about it, we have palms, the palms of our hands, of course, our front feet if we were dogs and cats. And the hind feet, that's you plant your feet into the ground. So you can try to think about it that way and see if that clicks a little bit more. So then, of course, dogs and cats and animals in general, they're not inanimate objects, so they have their very own left and right. So unlike our desk, if we're looking at our desk, the left side of the desk is our left side, the right side of the desk is our right side, no matter where we're standing. It always represents the same side as us. Dogs and cats, if we're looking straight at them, then it's opposite. So of course, this cat sitting in the bathtub, that is its right ear. And this sweet little cat named Moose, my favorite cat in the whole wide world, that's his left ear. Ringo, he was adopted at the college. That is his left foot. And that was a heck of a lot of words for directional terminology. So a little bit of practice. This cute little rat pushing a cat in a stroller, totally normal, see it every day. Not so much. So if we try to identify where that wound is on the body, we might say it's toward the dorsum on the lateral aspect of the thorax or the lateral aspect of the shoulder blade, the scapula. So identifying that it's on the lateral aspect and it's toward the dorsum would be key. And of course that it's on the left side, also key. And then we look at this cutie and he's got another little wound down on his foot. So that's his left foot. And it's on the dorsal aspect of his left foot. So we're going to move on and talk to essentially the basic units of life, and then eventually we'll make our way through the body systems. So if we think about the small components of life up until the large components of life, we've got cells, which are, actually I'll go through each of them on its own. So cells are the basic unit of life. They are specialized based on function. And of course, cells in the body are taken care of and protected by the body. Each cell relies on other cells in order to complete its function. They message to each other, they send receptors to each other. There's lots of communication in between cells. And just going back to the specialization based on function, examples of that, of course, are epithelial cells have a function to protect and cover the body. Intestinal cells might have specialized cells that assist in immune function. They might have cells that are responsible for exchange of micronutrients, etc., etc. So they're all specialized based on where they are in the body. And then we have tissues, not so much that tissue, although I could probably use one right now. They are bundleized, that's not a word, bundles of specialized cells. We have epithelial, connective, nerve, and muscle. Examples of epithelial tissue are, of course, epithelium, the skin that's around all over the body, that's protective, and its main goal is to cover and protect the body. Connective tissue, we're often talking about fat, cartilage, fascia. Nerve tissue is throughout the body, throughout the spinal cord, the nerves themselves. And of course, they transmit sensory information from the body to the brain, and then the nerve directs the body on how to act. Muscle, we have 3 broad categories of muscle in the body. We have smooth muscle, which is then further broken down. We have skeletal muscle, and we have cardiac muscle. Skeletal muscle is all throughout the body, a lot of smooth muscles throughout the body as well. And then, of course, cardiac muscle we only find in the heart, very specialized. And then we're working our way sort of to the base of that pyramid. Organs are groups of tissues working together with a common goal. Some of them are in pairs, and others work on their own or single. So if we look at organs that are in pairs working, of course, we've got the eyes, the lungs, the ears, etc., etc. Kidneys, of course, in the background there. And then others that work on their own, we have all the components of the gastrointestinal tract. They work on their own, but as part of a larger body system. So body systems are, of course, groups of organs working together. So we're going to break it down, and we're going to start to talk really in small terms. So we'll start with the cell itself. And before we start talking about the cell, we have to talk about homeostasis. Homeostasis is the state of equilibrium in the body. And essentially, it means good health. Homeostasis includes regulation of body temperature, of pH, of nutrients, fluids, essential gases, hormones, enzymes, etc. So homeostasis is the overall balance of all of these mechanisms in and out as the body maintains balance. So the example that your textbook gives you is that essentially homeostasis is like the little inputs that it takes to maintain yourself on the road. So in this case, the road is life. The car is the animal's body, and homeostasis is all the little ins and outs, all the little inputs that go into driving, avoiding roadblocks, turning on your I almost called it your flicker, your turn signal, etc., etc. So there are all those tiny little components that go into driving just to keep that car straight on the road, and you do it without even thinking for the most part. So homeostasis is very, very similar to that. An example of sort of homeostasis and how if one system doesn't work, then other systems stop working and the balance within the body is disrupted. So an example of that is congestive heart failure, which is a heart that's not pumping well. It has decreased cardiac input, and therefore the blood is pooling and getting backed up in the vessels. So that's where the congested part comes in of congestive heart failure. When the blood starts pooling, sensors in the vascular system and the central nervous system, they sense the low cardiac output and they set the body into fight or flight mode. When that happens, everything has to work harder. So fight or flight mode means that the body starts pumping everything. Unfortunately, this pumping action, this massive boost in pumps, it doesn't in fact increase the cardiac output. Instead, it exhausts an already poorly active heart and causes it to weaken even faster. So in the end, if the heart isn't working, but all the other systems are, the body still can't maintain homeostasis. So regardless, homeostasis always attempts to maintain the body in a state of equilibrium, but if there are broken parts, then the body won't be able to do that, and homeostasis just won't happen. So we're going to go down to cellular level, and for us, we're going to be talking about eukaryote cells. So prokaryotes are before the nucleus. So essentially 3 billion years ago, they're cells essentially that are similar to common day cells, but also quite different, and they are before the nucleus, so before the nucleus arrived. Eukaryotes are the ones that we're going to be talking about. That's the true nucleus. Bacteria versus viruses, just a little word about them. Fungus and bacteria are cellular beings. They can be seen under a light microscope. Viruses are essentially just parasitic, and they're too small to be seen with light microscope. So in those cases, we often use electron microscopes to get an idea of what viruses are doing. And the way viruses work, what was discussed in class, is of course they attach to cells, healthy cells, and they inject their DNA into the cell. And the injection of that DNA inhibits the cell's ability to function normally. So going back to eukaryotes, we're going to talk about those cells. When we're talking about that nucleus, it's the genetic material hub, and of course, one of the most important parts of the cell. Most cells have one nucleus. The exception is really large cells like skeletal cells and cardiac cells, and the number of nuclei is based on surface area of the cell. So a little bit of a reminder for terminology. Inside the cell we refer to as intracellular, outside the cell extracellular, and in the extracellular tissue we talk about that as interstitial. So looking at our cell, of course we know that animal cells do not have a cell wall, instead they have a plasma membrane. And the plasma membrane is smaller than microscopic membrane that maintains the contents within the cell. So it keeps the shape of the cell and maintains everything within it. And that should say plants, not plats. Plants, of course, have a sturdy cell wall. So this plasma membrane is constructed of globular proteins, phospholipids, cholesterol, etc. And cholesterol plays a fairly big role in maintaining the rigidity and the structure and the firmness of these cells. It is a flexible cell overall, flexible plasma membrane. It controls the movement of molecules in and out of the cell. And then we have the inner and outer layers. Little hydrophilic aspects are on the outer layer, and hydrophobic, a lipid bilayer, is in the middle, or on the inside of the cell, on the inside of the plasma membrane. And then, of course, suspended molecules in between the two layers are the fluid mosaic. So the two-layered structure of the membrane allows some molecules to come into the cell, but make it more difficult for other molecules to come in. So the inner and outer layers, there's fluid in between. The hydrophilic layer is on the outside, hydrophobic is on the inside, and suspended molecules in between the two layers create the fluid mosaic. The two-layered structure of a membrane allows some molecules to come into the cell, but make it difficult for other molecules to come in. So an example is that oxygen and carbon dioxide easily pass through An example is oxygen and carbon dioxide easily pass through the hydrophobic layer into the cell. Amino acids, proteins, and sugars do not readily pass through the membrane into the cell. And we'll talk about different mechanisms of action of how those molecules make their way into the cell when they're needed. So there are little integral proteins act as passageways to allow certain substances into the cell and others to stay out of the cell. So that's a nice little diagram on the left, and that's one way that materials enter into the cell. Glycoproteins and glycolipids are on the outside of the cell, and they create this sticky outer surface. The glycocalyx is essentially that overall, I'll show you what it looks like. Overall kind of fuzzy appearance to the outside of the cell. It's like an extra little layer, which is unique to every cell. It's like their fingerprint almost. The benefit is that the cells are able to recognize each other by their glycocalyx. Downside is that bacteria and viruses can also zero in on particular cells based on sort of that fingerprint characteristic of their glycocalyx. So what I'm talking about with glycocalyx is this is the cell, and then of course attached to the plasma membrane is the glycocalyx, which creates that fuzzy type appearance. And this is what it really looks like, essentially on an electron microscope. Carrying on with the plasma membrane, it engages in chemical signaling, and it also has binding sites that fit specific shapes. So a good example of this is opioid receptors. When we give an animal an opioid drug, we rely on the fact that they have these little opioid receptor binding sites on their cells, and when they connect, when it's like that missing puzzle piece and they connect perfectly, then that opioid is allowed to have an effect on the cell itself. So it's a specific shape, and it only fits that specific receptor. All right, moving on away from the plasma membrane, we'll talk about some other cell parts, and then we'll enter into organelles. So parts for locomotion, we have flagella, which is a tail for locomotion, and cilia, which is essentially thousands of flagella. So the video here, if you have the PowerPoint copy, you can click into it. It's just a nice little video with cilia and with flagella to show you the movement. Examples, of course, we'll find ciliated cells, typically ciliated columnar epithelial cells, all throughout the respiratory tract, and they have this brush border. Now, these ones aren't like detaching from other tissue and roaming around the body, but instead, these ciliated cells use their little brush border to expel dirt, debris, foreign objects from the respiratory tract. So this is sort of like the inside of the trachea, and then these are all those ciliated epithelial cells toward the inside of the trachea. And then, of course, an example of flagella is our friend the sperm. And they have a large flagella that helps them get around. So moving on, the cytoplasm, it's the inside of the cell, excluding the nucleus. And within the cytoplasm, the organelles can be found. Essentially, we used to think of the cytoplasm as a nondescript bag of gel with a few speckles. That's what we tend to see with light microscopy. But once we look at it through an electron microscope, you can see all the components of the cytoplasm and how interesting and how easily and quickly it can change. Now, the cytoplasm, this is a good picture here. So these are red blood cells. Looks like a lymphocyte with some cytoplasm here. Cytoplasm will change colors and consistency and increase in vacuolation, so holes, depending on disease processes in some animals. So one thing that we definitely do as you guys start to look at lab work is we always assess the cytoplasm to identify if it looks normal or if it looks toxic or if it's vacuolated, etc., etc., etc. So cytoplasms are very important to us with cell ID and identifying disease processes. Mitochondria is one of the largest organelles within the cell, and it provides 95% of the cell's energy needs. So we often label the mitochondria as the powerhouse of the cell. The number of mitochondria depends on the activity level of the cell. So as you can imagine, the heart that doesn't stop, it doesn't cease activity, so it does not stop beating, of course has tons and tons of mitochondria. It is also responsible for cellular respiration, so that's the intake of oxygen, yum, 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 oxygen, and, quote, the liberation, i.e. the excretion of carbon dioxide. So you can see our little Pac-Man there is eating the oxygen, and then, of course, having a bowel movement to poop it out. It doesn't quite work that way, but you get what I'm saying. So then we have these guys, the ribosomes. We'll talk about these a little bit later when we talk about protein synthesis and DNA transcription. They're the most common organelle in the cell. So 2 subunits containing RNA and proteins. They are smaller than the mitochondria. They move around the cell, and they're heavily involved in protein synthesis. So they actually go to where they are needed within the cell. Essentially, they can produce, put together pretty much, soluble protein for intracellular use, and they of course also produce proteins for extracellular use. So they're really, really important in the functioning of the cell. Then we have the endoplasmic reticulum. We have the smooth and the rough. The smooth, synthesizing and storing lipids, elimination of drugs, breakdown of glycogen to glucose, and we'll talk about that later on with digestion, that whole process. And then rough aids the production of proteins. It aids the mitochondria, and of course they're covered in ribosomes. The rough is covered in ribosomes. So just going back, to clarify, the endoplasmic reticulum, the key role in the endoplasmic reticulum is protein and lipid synthesis. And then we have the Golgi body, well, now called the Golgi apparatus, and I need to start conforming and calling it the right word. So the Golgi apparatus, this little guy receives new proteins from the endoplasmic reticulum. It modifies molecules, and then it plays a role in sugar attachment and carbohydrate synthesis. So essentially, it's taking pieces in, modifying them, and then sending them back out. So we like to call this one a little miniature modifying, packing, and distribution center. Lysosomes are kind of cool. They are formed by the Golgi apparatus. They're also known as the stomach. I like to think of them as kamikaze bags, and they can undergo autolysis. So normal functioning, they break down food molecules into usable units for cell parts. They digest and get rid of intracellular debris, foreign invaders, and bacteria. If the lysosome rupture, the enzymes within it can destroy the cell. So really similar to the stomach, the actual organ, the stomach. If starving cells, I'm reading my notes all wrong. So if other cells are starving, or if other cells are, or if that cell in particular is not doing very well, then lysosome can cause self-destruct. So it can cause this process called autolysis, whereby essentially it destroys itself, which in turn means that the cell gets destroyed, and all of the organelles from that cell get essentially broken down, absorbed, and the nutrients that are lost by that exploded cell can get used by other cells and cell components. So the little kamikaze, i.e., kind of like suicide bags, they're ready to explode. They are the self-destruct. And then we have these guys, peroxisomes. I think these are super cool. Liver, they're mostly found in the liver and kidney, They're not produced by the Golgi apparatus, and they're involved in detoxification of compounds, such as alcohol, formaldehyde, removal of free radicals, which are not good for cells. So essentially, they have enzyme production, peroxidases and catalase. Peroxidase converts free radicals to H2O2, and catalases reduces the H2O2 into H2O, so into water. So what I like to think of it, because they're detoxifying, they take harsh substances and they turn them into usable substances. They are the detox little organelle. So they take alcohol, such as this fabulous selection from Bellwoods Brewery in Toronto. They take alcohol and they turn it into water. It's not that straightforward, but we should all love these little guys, our little peroxisomes, when it's weekend. All right, the nucleus. I don't know how to put this, but I'm kind of a big deal. The nucleus is a giant deal. It has two main functions. So it maintains the hereditary information of the cell, and it controls the organelle activity through protein synthesis. So the nucleus essentially has the DNA, and the DNA has all the blueprints of how to create all the proteins that are needed by the body. Essentially, it's the CEO. It just directs everything, tells everything what to do and how to do it. Typically, it's the largest organelle in the cell, so aside from the nucleus, right? We're not talking about, or aside from the nucleus, brain fart. Ignore that. It's the largest organelle in the cell and the main center of the cell. It directs and controls the actions of other organelles. Most cells have one, some cells have more than one nucleus. We'll talk about that when we start talking about cancerous cells and they start to undergo abnormal replication. When staining cells, so when we're looking at them through a laboratory setting, most nuclei stain really dark if you're using a stain that's set up for nuclear material, such as a Wright's or a Gemsa stain. So here's our nucleus in the middle of the cell, and it's not considered part of the cytoplasm, okay? So it is separate from the cytoplasm. So interesting fact, mammalian red blood cells don't have a nucleus. They die after 3 to 4 months. Avian and reptiles have red blood cells that are nucleated. So they tend to live much longer in the body. So mature mammalian red blood cells, they have no nuclei, like I mentioned, because the nuclei are removed from the cells during their development in the bone marrow. So these cells, we call them enucleated, so without a nucleus. Without a nucleus, they can't divide, they can't make protein or enzymes, and they can't really repair themselves as they start to age, and that's why they only last 3 to 4 months in the bloodstream. Now, sometimes we do see mammalian red blood cells that have a nucleus in them, and it can be various stages of nucleus. So if that's the case, we have to note what type of cell that is. So if it's a ruberocyte or metaruberocyte, which are immature red blood cells, And we have to be very careful about noting that on our laboratory results, because quite often what that means, there might be a systemic anemia, and the body's pumping them out of the bone marrow before they're ready. So they're sending them out because the body identifies a loss. So that's another example of homeostasis, in that the body is trying to correct what's imbalanced. So there's a loss of red blood cells. The body's going to push the bone marrow to pump out more red blood cells. So for this reason, in general, about them lasting such a short amount of time, the supply of vital molecules in mammalian red blood cells allow them to survive in circulation for 3 to 4 months, like I said, unlike those that can survive for longer periods with the nucleus, such as avian and reptile. So the nucleus, we'll talk in regard to the nuclear envelope or the nuclear membrane. There's the nucleoplasm, which we won't really discuss much at all. Chromatin we'll discuss as a discussion about DNA and protein synthesis. And then the nucleoli, which is central within the nucleus. Overall, nuclei, which is the plural for nucleus, nuclei are complicated. The nuclear envelope, or the nuclear membrane, essentially separates the nucleus from the cytosol, and the cytosol is that gel that makes up the cytoplasm. It contains pores or channels that aren't very selective, okay? So they can let a lot of things in, let a lot of things out. I like to call them lazy. So I like to call the nuclear membrane quite lazy. And of course, the nuclear membrane moves molecules from the cytosol into the nucleus and vice versa. So it acts as a huge communication pathway. Chromatin is the genetic material prior to cell division. It's the hereditary portion of the cell. Very, very important. And we'll talk about chromatin in a little bit when we start talking about protein synthesis and cell replication. So this is what the nuclear envelope looks like. It has little pores all throughout it, which are not overly selective. Okay, so a little bit about chromatin. We've got a chromosome, which is the full formation, ready for division. And then we have the looser chains, the thinner chains, so thin chains of genetic material, which are called chromatin, and of course they are made up of DNA. In class, if you're working with me through a lecture, there's a little hyperlink here, which is really great to just explain a little bit more about chromatin and DNA and how DNA is formed. It's a good refresher. And I also put it in my playlist, so if you're not in the course that I'm teaching, you should be able to find that link in my playlist. So essentially, DNA contains all the important information on how to create thousands of proteins in the body. And those proteins are what essentially form together to create new cells, to create organs and tissues and everything. But it doesn't go ahead and make all the protein. Only some of the DNA is expressed, and therefore only specific proteins are synthesized by DNA in each cell. Chromatin's loosely scattered throughout the nucleus, except during times of cell division when it clumps into dense chromosome formation. Okay, so chromatin normally just hangs out, scattered in the nucleus, and then you'll see it clump up and become more dense during cell division. And then we have our nucleoli. Two functions contain the DNA that govern the synthesis of ribosomal RNA and the creation of ribosomal subunits. It's exported from the nucleus and assembled in the cytoplasm into functional ribosomes. Sorry, the functional RNA is. I'm losing it. I'm getting sick. Okay, and then we'll talk a little bit about water in cells. So cells are 70% water, roughly, and essentially it's similar to the body. The water in the cells helps to maintain the shape and integrity of the cell. Enzymatic reactions and functions, water can always assist with these depending on the reactions itself. Cellular fluids contain many charged particles or ions. And then when we're talking about charged particles, cations are positively charged, anions are negatively charged. There are a few memory tricks that came up in lecture today. So cations, if you look at the way cat is spelled, T is kind of like the plus, the positive symbol. So if you look at that, it's positive ion. So cation is positive ion. Anion, I always think of an as without, so that's negative. And I forget the other memory trick already that somebody told me. But the one I like is a cation, is an ion with positive charge, and it's the cutest ion ever. This was a nice little cartoon that was on the internet. I like that the best. So moving on with water in cells, the movement of ions within the cell is often dependent on the charge of the ion. All ions are termed electrolytes, and salt is an example of an ionic compound, and along with other electrolytes make up most of the charged particles in a solution. So electrolytes are capable of conducting an electric current. Therefore, they can have huge implications for certain organs if an imbalance occurs. So this is where it becomes clinically relevant and clinically applicable for veterinary technician students, is to note that we don't play around with electrolytes. We don't take them lightly. If an animal is very, very low on electrolytes, or if we've given them too many electrolytes, they get into this system of an imbalance, and they can have major cardiac implications, muscle implications, etc., etc. So we don't mess around with electrolytes, but we always need to replace them and create balance with electrolytes. That's our goal. Solutions with an excess of positively charged ions are termed acidic, and solutions with an excess of negatively charged ions are termed basic. The more hydrogen in a solution, the more acidic the solution, and of course pH is a measure of the concentration of hydrogen ions in the solution. When we're talking about mammalian blood, pH is typically 7.35 to 7.45. If the pH is less than 7, then the enzymes are not functioning effectively, because of course 1 to 7 is acidic, that's considered acidic, and 1 to 7 is acidic, and then 14 is basic. And either ends of those spectrum are not ideal, because it means that the body is not maintaining homeostasis. So going on a little bit about electrolytes, electrolyte balance is key to normal bodily functions and safe bodily functions as well. Electrolytes affect the amount of water in the body and the acidity of the blood, the muscle function, and other hugely important processes. Hormones produced in the kidneys and the adrenal glands often direct the concentration of electrolytes in the body. And when disease processes take hold, electrolyte balances are disrupted. So we often have to add potassium. If an animal is lacking in potassium, we'd have to add it to their IV bag, or we might have to add bicarbonate, likewise if they're slightly acidic in their blood, to try to create that balance again. It's all really important. The main thing here is that you need to know the implications of giving too much or an animal not maintaining their electrolytes. Just going back when I was talking about the hormones that direct the concentration of electrolytes, a common medical process is Addison's disease, and that's an adrenal gland disease that, of course, has implications for direction of electrolyte concentration. And in that case, we get an inappropriate sodium to potassium balance. So we get a decreased sodium, an increased potassium. And then my dog had that, and of course she ended up with muscle tremors, she ended up with a cardiac episode, all sorts of huge issues. Her body essentially just gave up, and in the end she couldn't maintain blood pressure until we got the source. We figured out what was wrong with her and got it fixed with medication. So an imbalance of electrolytes can be a huge deal for the body and can definitely put them into a situation where death is around the corner. All right, so moving on to the membrane processes, because we have to talk about how substances and molecules get inside the cell or how they leave the cell. So the selectivity or the permeability of membranes often defines the movement of molecules and substances required for cell maintenance. It's often dependent on energy needs. We have active processes such as endocytosis and exocytosis, and passive such as diffusion, facilitated diffusion, osmosis, and filtration. The energy form required for active transport of these molecules is ATP, so adenosine triphosphate. So an example, the movement of amino acids, gases in solution, hormones, and vitamins may or may not require energy in order to be transported through that plasma membrane and into the cell. And of course, when we're talking about active processes, we're talking about using energy, needing energy. And when we're talking about passive, we're talking about no energy required for that to happen. So we'll talk a little bit about diffusion and osmosis, etc. But essentially, diffusion is based on a concentration gradient. It's the passive movement of molecules in a fluid by kinetic energy. So if the fluid is warmer, then the molecules will warm up, they'll be more active, and that's our kinetic energy. Generally, these molecules move from areas of higher concentration to areas of lower concentration, and the diffusion of molecules is increased with heat, like I just said. Okay, so this is a perfect example of diffusion, whereby we're adding a solute, such as food coloring, into a solvent, which is water, and the food coloring is moving around from its area of high concentration to low. And then we have facilitated diffusion. So this is when we have large non-lipid molecules require the attachment to a carrier protein to pass through the membrane. So essentially they need like a bouncer to let them in, or a gatekeeper. Particularly, the molecule carrier protein binding sites are required, and some alteration in shape is facilitated so that certain molecules can actually pass through the membrane and into the internal cell environment. So an example of this is movement of glucose from the gastrointestinal tract into muscle and into fat cells. It requires a concentration gradient, so the facilitated diffusion overall requires a concentration gradient as well. And I like to think of it as, you can think of it as like a bouncer walking you into the bar, or a gatekeeper who opens the gate and sort of walks you into the pathway. I like to think about getting on a spaceship and that you can't really get into the stratosphere beyond just on your own. So you have to enter this carrier molecule, or this carrier protein, in order to get to that next level. So molecules would have to enter the carrier protein in order to get inside the cell. And I especially love this example because there's a giant cat on the spaceship. So then we have facilitated diffusion, and each cell has a limited number of these carrier proteins. Generally, this process is mediated by the number of carrier proteins available. I don't know why I said, and then we have facilitated diffusion. We're talking about the same thing. My mistake. So you're welcome to watch that video as well, just for a general overview of how facilitated diffusion works. And then we have osmosis, which is another passive method for getting through the plasma membrane. It's the passive movement of water through a semi-permeable membrane into a solution where the water concentration is lower. The membrane in this case is semi-permeable only one way, so the water can go one way but not back the other, and the water continues to move until an equilibrium is reached between the outside and the inside of the cell. Its opposite to diffusion is the movement of the solute, and osmosis is involved in the movement of the solvent. So here's an example. This is diffusion. It's, quote, like the salt is moving to an area of lower concentration, so it's moving through the water into an area of lower concentration, whereas osmosis, the water is moving to an area of higher concentration to dilute it. And now we'll talk about osmosis as it relates to tonicity. So if the concentration of extracellular fluid is greater, i.e. hypertonic, so that's higher concentration, then the lower concentration inside the cell, hypotonic, that means lower concentration, then the fluid will move from the cell into the extracellular area. An example are RBCs, so red blood cells in hypertonic blood will become crenated because the fluid from within the red blood cells sees that there's concentration on the outside of the cell, so the fluid will want to dilute that concentration. So essentially your red blood cells deflate, the fluid is escaping from the red blood cells, and it enters into that area of higher concentration. And when red blood cells get all shriveled up, they're called crenated. Conversely, if the fluid in the cell is less concentrated than the extracellular fluid, water will flow into the cell. An example of isotonic solution. So isotonic is well balanced, essentially. The concentration is balanced. And an example of an isotonic solution that we give to animals is sodium chloride. So actually, it's sodium chloride 0.9%. The difference between osmotic pressure of blood and the osmotic pressure of interstitial fluid or lymph is called oncotic pressure. And this is an important force in maintaining fluid balance between the blood and lymph vessels in the fluid in the surrounding tissues. So in some disorders, the balance of fluid between these two spaces is disrupted. And of course, one might become more concentrated than the other, particularly if there's a decrease in the number of protein molecules in the blood plasma. Starvation, liver failure, and intestinal disorders, for example, can cause the levels of plasma proteins to decrease. And if the levels become too low, the fluid will move out of the vessels because now the space outside is more concentrated. So the fluid will move out of the vessels and into that other space because it's deemed more concentrated, hypertonic. So in those cases, the fluid is escaping from the vessels and it's moving into other areas within the body. This in particular is called ascites, so it's fluid filling up the abdomen. Okay, now ascites is really common with end-stage liver failure and also with starvation. Fluid always finds a pocket. That's one little thing to remember. So based on that, fluid always finds a pocket to fill up. So these are our RBCs. So before I was talking about crenated RBCs. So this is a normal RBC. They're kind of like a dish or a saucer. They have a central pallor, so like a divot in the central pallor. In this one, in C, they are overinflated. So that is when you've got, the solution outside the cell is less concentrated than inside the cell. And then the fluid will squeak into RBCs and overinflate them. And then D there, you have crenated. So when the fluid outside the cell is more concentrated, it's hypertonic, then fluid will escape out of your red blood cells and they'll become crenated, so all shriveled up. Filtration is based on a pressure gradient. So this is another method for getting molecules in and out of the plasma membrane. Liquids will be pushed through a membrane if the pressure on one side of the membrane is greater than the other. The pressure, or the force, that pushes the fluid is known as hydrostatic pressure. In animals, blood pressure is created by a pump, so the heart, through the blood vascular system. The kidneys function as a filter using the similar mechanism, so all through filtration. Now, exceptions to the rule with filtration, areas of the body that are specialized to maintain high levels of pressure and not allow for fluid to escape are, of course, the aorta, the carotid artery, and arteries in general because they maintain high pressure. And the kidneys also have areas of really high pressure. And then we start getting into active transport. So an active process is needed to move molecules through membranes that normally don't allow... Oops, my cat's on the computer. Okay, gotta go. Silly kitty. So I'm going to repeat that. An active process is needed to move molecules through membranes that normally don't allow passive passage. So either the molecules to be moved are not lipid soluble, and the membrane is, the molecules to be moved are too large for the pores of the membrane, or the molecules may be on the wrong side of the concentration gradient. So we'll talk about some active transport mechanisms, all of which require ATP in order to happen. So it does not require a concentration gradient. Unlike facilitated diffusion, it's actively using ATP, so energy. In general, the body's not trying to equalize anything. The primary use of active transport involves movement of electrolytes such as sodium, potassium, chloride, magnesium, and others throughout. So here's an example. Endocytosis occurs when large particles, liquids, and even whole cells are taken into the other cell. Molecules are normally moving from extracellular areas to intracellular areas. So this is when the cell takes that molecule or cell or bacteria into the cell. So if they're taking in another cell, or if they're ingesting a foreign invader, such as bacteria, that's called phagocytosis. So right there, phagocytosis. And right here, essentially white blood cells undergo a lot of phagocytosis. That's one of their main roles. So if there's bacteria sitting between cells or in the tissue, then white blood cells will go in and go through a process of endocytosis, whereby they take in that bacteria or foreign invader into their cytoplasm for destruction. So this one here, we've got a nice big cell. There's its nucleus, its cytoplasm. And then within the cell cytoplasm, we've got some rod bacteria. And then it looks like we have pairs of cocci on the outside of the cell. And then just looking at this one too, that's the glycocalyx. So that's that sticky outer layer that acts for signaling, but also as the fingerprint for cells. So macrophages are a version of a white blood cell, and their whole role in life is to engulf foreign invaders or engulf dead cells and destroy them. And I love this. This is the Amoeba Sisters. They have a whole channel that really breaks down a lot of these concepts, but this killed me, just cracked me up. Dear Diary, what a Monday. Today I was engulfed by a giant cell out of nowhere. I love it. Anything with a cartoon. So carrying on, exocytosis, exocytosis involves the movement of molecules from the intracellular to the extracellular. So it's the opposite of endocytosis. The process is used to remove cellular wastes, so that's excretion. And if the molecules to be removed from the cell are manufactured by the cell, then the process is known as secretion. Most secreted substances are packaged in little vesicles, and eventually those vesicles, which are essentially little packages, fuse with the cell membrane of another cell, and the contents are released into the extracellular fluid. So excretion, we're talking about discharge of wastes from the cells, and secretion, we're talking about products that are created and then released by those cells. So a good example of that is the secretion of hormones, secretion of insulin, secretion of mast cells, secretion of histamine by mast cells. And then here's another happy little video to watch if you have time. And then just the tiniest of tiny little bit about membrane potentials. Just to note that the distribution of charges on either side of the cell membrane forms a membrane potential, so forms a voltage. Normally, the inner side of the cell membranes exhibit a negative potential. The cytoplasm and extracellular fluid has no charge, but it does carry ions. And note that active and passive membrane processes result in higher positive charge outside the cell than inside. So we all talk about but just overall potentials when we're talking about ions and voltage. So we will talk about this concept much more when we start talking about cardiac function.